Hi everyone, welcome back to Studying with Zahra. Today we're going to be doing Lipids, AQA, AS level / A level, and this is the ultimate guide. We're going to be finishing it in one lesson, and this is part of biological molecules. Now, just to begin with, some really important things to bear in mind is that lipids are made up of carbon, hydrogen, and oxygen, which are the same atoms that we know carbohydrates are made of, and also proteins are made of, except proteins have some extra elements such as nitrogen, possibly even sulfur. We're going to learn more about that in the next lesson. Now, the proportion of oxygen to carbon and hydrogen is smaller than in carbohydrates. They're insoluble in water, and they're soluble in organic solvents such as alcohol and acetone. The main group of lipids are called triglycerides and phospholipids. They sound a little bit confusing right now, but I promise we'll go over it. Now, I've made my little notes on the margins, but essentially fatty acids vary in length and bond structure, but they always end in a carboxylic acid group. And if you don't get this right now, don't worry, I'll explain it soon. And a single bond between carbon to carbon is a saturated fat. You should know this from your GCSEs. And a double bond is an unsaturated fat, because essentially what saturated means is it has the most carbon-carbon bonds possible. And so when the bonds are single, then that means that it can have the most carbon-carbon bonds possible. And when the bonds are double, it can't. So therefore it's unsaturated. Now, lipids aren't polymers, unlike the carbohydrates that we learned before, like starch, cellulose, because we know the polymers are made up of a specific monomer that is repeated multiple times, essentially. And lipids aren't made of one specific monomer. They're actually made up of two. So they're made up of fatty acids and glycerol, which are not the same, and so we don't call them polymers. So the rules of lipids. Now, I made little drawings in the margin just to make it a little bit more interactive. But essentially, lipids are a great source of energy, and you need them in your diet. Other than carbohydrates, they provide so much energy. They are essentially just used up in respiration to produce or to release ATP. Next up is waterproofing, and you may know how leaves have little waxy cuticles. So the waxy cuticle is essentially made up of a lipid, and this prevents excessive water loss of the leaf. We also have a little bit of oil on our skin. Sometimes your face might get a little bit oily and whatnot, and that is essentially just lipids. Again, prevents water loss. You'll know that if you strip your skin too much, so for example, you wash it too much, it actually does tend to get oily because your skin is trying to prevent itself from drying up. Insulation. Now, I feel like this is pretty straightforward. Insulation just essentially means to keep you warm and to retain the body heat. So a lot of fat stores are found in the skin, so that when it's a little bit cold, you don't feel too cold and your body temperature is regulated. Lastly is protection. Fat is stored around delicate organs, so maybe like the kidneys, and this just ensures that those specific organs don't get damaged. They are cushioned, they're protected. So next up, we're going to be going over triglycerides. Now, triglycerides are so called because they have 3 fatty acids and a glycerol. I feel like it's pretty straightforward. Now, each fatty acid forms an ester bond with the glycerol in a condensation reaction. And so, in order to go from fatty acid and glycerol to the triglyceride, we need to undergo a condensation reaction. 1 molecule of water is produced. Now, you have to bear in mind that for every single fatty acid that bonds with a glycerol, that is 1 condensation reaction. So if I have a triglyceride, that means that I have 3 condensation reactions. If I have a diglyceride, I have 2 condensation reactions because I have 2 fatty acids and a glycerol. And if it's think of monoglyceride, then I have one fatty acid and one glycerol, so that's one condensation reaction. And hydrolysis is essentially the opposite, so a water molecule is used up. The ester bond is broken down, and you get back to your reactants. So it's pretty straightforward. We've done really similar stuff in carbohydrates. Now, how much is left? Let me just check really quick. Not much. Now, so triglycerides can be different, but glycerol is always the same. So only the fatty acid varies. Essentially what this means is triglycerides are made up of your fatty acid and your glycerol. Triglycerides can be different, but we know that for every single triglyceride, the glycerol is always going to be the same, which means that one thing has to be changing, and that thing is the fatty acid. So the glycerol molecule, blah, 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 always the same. There are 70 types of fatty acids, all end with a carboxyl group. If it has a carbon-carbon double bond, then it is unsaturated, and if it doesn't have a carbon-carbon double bond, it's all single bonds and it's saturated. One double bond is monounsaturated, multiple is poly, pretty straightforward. And so yeah, I feel like, again, pretty straightforward. Now we're going to be looking at the structure of triglycerides and how those relate to their properties. So initially they have a high ratio of carbon-hydrogen bonds to carbon atoms and are therefore an excellent source of energy. Essentially what that means is, over here you can see that for every carbon atom that I have roughly 2 hydrogen bonds. So there's almost double. And the carbon-hydrogen bonds are a great source of energy. Triglycerides have a low mass energy ratio, so they're really good storage molecules because they're very compact. And this is especially great for animals that have a lot of movement, essentially, like us humans, because it means that we can store a lot of fat in a tiny little volume, so maximum the amount of energy. Being large non-polar molecules, they are insoluble, they don't affect the osmotic gradient of cells, and they release water upon oxidation. Therefore, they are an important source of water, essentially, for living organisms, specifically in dry, arid environments. Once again, just to go over it briefly, high ratio of carbon-hydrogen bonds to carbon atoms means that a lot of energy can be released. Secondly, they are very compact. A large mass of fat can be stored in a small volume, meaning that especially for moving animals, it's great because it's less mass to carry, and they're also a great source of energy. Thirdly, they release a water molecule upon being oxidized. I need to remember this. God, so they release a water molecule upon being oxidized, hence making them super useful for plants in dry, arid environments as well as animals. And then last but not least, they are not going to affect the water potential of the cell because they're non-polar and they're also insoluble, which is pretty straightforward. Next up, we have phospholipids. Phospholipids have one fatty acid replaced with a phosphate molecule, so you're going to have 2 fatty acids, 1 phosphate, and then last but not least, your glycerol. The fatty acid molecules repel water, and the phosphate group is attracted to water, thus making it hydrophilic. You have to state what hydrophilic means in the exam to get the mark. And the fatty acids are actually hydrophobic. Now this creates a polar molecule that results in the phospholipid turning into a little phospholipid bilayer within the cell, creating the cell surface membrane. So essentially what that means is your phosphate heads, are toward the outside because they are attracted to water, and then toward the inside we have our fatty acids because they repel water. So they're toward the inside, and it just creates a little bilayer. So the hydrophilic head and then the hydrophobic tail. Now, this means that phospholipids are polar, and that's just our little phospholipid diagram. Glycerol and fatty acid is joined in a condensation reaction. This is known as esterification because an ester bond is formed. The structure of phospholipids is related to their properties. So they are polar, they form a bilayer, and it essentially just creates a hydrophobic barrier. So, yeah, and the phospholipid structure allows them to form glycolipids by combining with carbohydrates at the cell membrane, which is important in cell recognition. So essentially, the phospholipid bilayer is crucial because it creates a hydrophobic barrier. This enables regulation of what goes inside and outside the cell. And by combining with carbohydrates, you can form glycolipids, which are essential in cell recognition. Last but not least, test for lipids is the emulsion test. So essentially, you add to your test tube your sample, your ethanol, give it a good shake, and then add in some cold water, and a cloudy white, or you could say like a milky color, just indicates the presence of fats. That's pretty much it, literally. And then we have proteins, which we're going to do in the next lesson. Anyway, thank you guys so much for watching. I'll see you guys next time, and I hope you enjoyed. Please let me know if you have any more recommendations.